All right, everyone. Thank you very much for coming to our second CDCR seminar. It's my pleasure to introduce Dr. Tobias Berg today, an associate professor in the Department of Oncology and an associate member of the Department of Biochemistry and Biomedical Sciences, and also the Medical Sciences Graduate Program. He's a member of the CDCR and is leading the center's translational oncology program, facilitating collaborations by acting as a molecular bridge between clinicians at the Dravinsky Cancer Center, JCC, and basic cancer researchers affiliated with the CDCR. He holds the Boris Family Chair in Leukemia and Hematopoietic Stem Cell Translational Research, and is the lead on the HHS McMaster Cancer Research Stem Cell Bank. The focus of Dr. Berg's research is on understanding determinants of treatment response in acute myeloid leukemia, AML, and on developing novel treatment approaches based on this understanding. His group studies the role of epigenetic regulators in AML and their interplay with regulators of lineage-specific differentiation, metabolism, and immune recognition, using functional model systems in vitro and in vivo, single-cell sequencing analysis, as well as functional metabolic studies. His program's goal is to identify novel treatment combinations and develop maintenance strategies to prevent relapse in AML after allogenic stem cell transplantation. Dr. Berg is also a hematologist at JCC, where he treats patients with hematological malignancies. Sorry, us chemists don't have all these long words. And is a member of the stem cell and cellular therapy program that offers autologous and allogenic stem cell transplants, as well as chimeric antigen receptor T cell, CAR T cell therapy. I know that one. Dr. Berg received doctoral degrees from the University of Freiburg, Germany, and from the Goethe University, Frankfurt, Germany. Without further ado, it's my pleasure to introduce Dr. Berg to the podium, where he will present epigenetics and AML. All right, sir.
Thanks a lot for the kind introduction. Thank you very much for giving me the opportunity to present in this seminar. Actually, it's the first one from here after an external speaker last time. So, and it's great to be part of the CDCR of the new center. I'm going to present to you today some of the research work that's going on in my group. Just a few disclosures at the beginning. And after that, I will get to my presentation by starting with an introduction on AML. AML is actually the most common acute leukemia in the adult population. Despite that, it's still a quite rare disease, but it's an important model disease because many of the concepts that we have about cancer treatment have originally been discovered and are still being discovered in this disease. The disease arises on the level, usually either of a hematopoietic stem cell or of a multipotent progenitor cell by the acquisition of mutations, where then the cell transforms into a leukemic stem cell that has the capability to self-renew, but has unfortunately lost the ability to differentiate into normal blood cells. And these undifferentiated cells that we call blasts, this is a picture of one of those, will then grow and outgrow the normal hematopoietic cells in the bone marrow so that these can't be formed anymore. So that's why many patients with this disease present actually with symptoms of either anemia or thrombocytopenia, so either with signs of bleeding or symptoms of anemia. Despite the disease looking relatively homogeneous, it's still a very heterogeneous disease. And in particular, it's not just one disease, but in particular through molecular studies, involving next generation sequencing, it was recognized that actually AML is not just one disease, but it's many, many different diseases. that are usually characterized by the co-occurrence of different mutations and patterns of these mutations. These mutations determine many things. They determine if the disease responds to treatment. They determine also potentially mechanisms how we can treat the disease. It's therefore very relevant to look at these mutations, and they more and more play a role also in treatment. Even though this disease is a lot of different subgroups of diseases, the treatment for this disease has for a very long time been very, very homogeneous. So because every patient essentially who could tolerate that treatment got one type of chemotherapy, which is called 7+3. This treatment regimen is about as old as me, and it's been, it consists of two different drugs. One of them is called cytarabine, and the other one is called daunorubicin. Daunorubicin given on 3 days and cytarabine given over 7 days. Our goal then with this treatment is to get rid of the blast cells in the bone marrow, or at least get rid of most of these blast cells in the bone marrow, and then allow the bone marrow to grow, or the normal cells to grow out again. So that going from this very homogeneous monomorphic picture, we'll go again to this like very heterogeneous, colorful picture of the normal bone marrow. Nowadays, we have treatment or additional treatments that we can add to this, which have shown some improvement in some subgroups of patients with AML. But overall, even with this type of chemotherapy, it's usually not possible to cure patients. But many patients for a cure will require another treatment that is called an allogeneic stem cell transplant. This is a transplant from a different person. So in order to do this treatment, you will need a stem cell donor. The treatment is not just the stem cell transplant, but it also involves intense chemotherapy that is given as a so-called conditioning regimen. So after this conditioning regimen, the stem cell product is then infused into the recipient, where then after a while, these new stem cells will grow out and will then give rise to the blood cells of the different lineages. It's important to notice that this stem cell product is not a pure stem cell product, but what we're actually infusing is a mixture of many different cells, many of which are immune cells. So they contain also cells like, in particular T cells, but also NK cells that then can recognize the leukemia and start fighting against the leukemia. So this type of treatment, it's not just this kind of high-dose chemotherapy that then requires the rescue with normal stem cells, but it's actually an immunotherapy. It's actually probably the first cellular therapy in that sense. The stem cell transplantation program here in Hamilton has grown quite a bit. So last year we performed about 100 stem cell transplants, and we're probably getting even above that number this year. So the stem cell transplant program has been quite successful, and this can also for many patients be a successful treatment. However, it has still to be noted that this treatment unfortunately still fails in some patients, or actually a significant fraction of patients. And when you ask the question, why does the treatment fail? And this is really an analysis from the CIBMTR, so the international registry that looks at stem cell transplants. The most common reason why this stem cell transplant fails is really relapse. So understanding how relapse arises, in particular in the setting of stem cell transplant, will be a very important thing to understand how we can make treatment better. So one of the other aspects is that stem cell, or the allogeneic stem cell transplant, has a limitation in the patients that it can be given to. It's a quite intense treatment. So in particular for older patients, we long didn't have very good treatments. Recently, just like last week, one of those treatments has been approved though, and this treatment is called venetoclax. So it's given in a combination. What venetoclax does, it's an inhibitor of BCL2, so it targets BCL2 and then thereby enhances apoptosis. It also does some other things that we'll get to later on, but the combination of Vidasa, so azacitidine, which is actually an epigenetic treatment, and venetoclax has led to an improvement in patients with this disease. However, despite that, there is still a lot of chemoresistance, which you can see here when you look at those survival curves from the original trial that led to the approval of this medication. So one of the questions that we're following up in the group is really, how can we prevent relapse and how can we overcome treatment resistance? And Pradhani, a master's student in the group who has just recently graduated, has looked at the role of the microenvironment in this context. And why was that interesting? When we look at the venetoclax, it's not just an apoptosis inducer, at least in these leukemic cells, but it has been found in studies that in particular have been published by the group of Craig Jordan, that that the leukemic stem cells have a very specific pattern or metabolic pattern or pattern of metabolism. So they use a lot of oxidative phosphorylation and they depend very much on this. And also the source of metabolites that they use for their oxidative phosphorylation is specific in the fact that they use a lot of amino acids to fuel their need for oxidative phosphorylation. Leukemic stem cells then, when they treated with venetoclax, they then have a reduced oxidative phosphorylation. This is mostly by targeting this mechanism. And when you're then looking at resistant cells, this mechanism doesn't play as much of a role, but they can start using fatty acids to fuel these processes. And this then led to our question, which is essentially what is the role of the microenvironment there? Because in the microenvironment of these cells in the bone marrow, there are a lot of adipocytes. The adipocyte is actually, under usual circumstances, the most common cell in the bone marrow. So the question was then, what could be the role of adipocytes and other cells in the microenvironment? And our comparator were really bone marrow mesenchymal cells, mesenchymal stromal cells. What are the contributors of those to the resistance to this treatment combination? And in order to understand that, or in order to investigate that, we were interested in also studying metabolic processes. And one tool that we have available in the lab to study these metabolic processes is the Seahorse metabolic analyzer, which essentially measures pH as well as oxygen in proximity to the cells that we plate. And by that can then provide a measure of both oxygen consumption rate, but also extracellular acidification rate, which are surrogate measures for glycolysis as well as oxidative phosphorylation. So the experimental design for these experiments was then to use or to differentiate bone marrow stromal cells into adipocytes or to leave them as they are, culture them with AML cells, and then analyze what is the impact of that on the growth of these AML cells and on their metabolism. When looking at the growth of these AML cells, we had a bit of Pradhani had a bit of a surprise that almost endangered her project a little bit, because instead of growing better, they actually grew worse, which is a bit inconsistent with which has been previously published and what we looked up in other publications, but that's sometimes how it is. So we were not too discouraged by that. The other thing is that even their viability was a bit reduced. Still, when we measured their metabolism, they had about the same basal respiration rate when measuring with the Seahorse metabolic analyzer. But these cells that were in proximity to the adipocytes had a higher maximum respiratory capacity. So they had a higher maximum respiration rate. They also had a slightly higher basal ECAR. So, and when we looked at what's going on with a normal when we looked in a patient sample, we saw actually similar patterns. So again, a reduction in proliferation, but still an increase in this maximal respiration rate. And this is an interesting phenomenon because this could be essentially showing us that there may be a bit of a selection going on for these leukemic stem cells or for cells that may have resistance. And that's why the next set of experiments really ask the question, what happens if we treat these cells that are in proximity to the adipocytes with this combination of Vidasa and or azacitidine and venetoclax? So, and when Pradhani did those experiments, she saw that to our surprise, the viability of these AML cells, in particular when they were treated with this venetoclax alone, was actually protected from the effect of the venetoclax. And even in the combination, we saw some protection in these cells. There was also quite a significant protection regarding viability. And so we further wanted to understand what the contribution of these metabolic processes is to that, but unfortunately, we didn't really see a difference. So we then went back and looked again at the mechanism, and one of the mechanisms that could cause resistance to this BCL2 inhibition is an upregulation of MCL1. So Pradhani studied the expression of MCL1 in these cells, and you can see that in the presence of the adipocyte, these AML cells, these were HL60 cells, actually upregulated MCL1. So this is one way probably how this mechanism works. It may not be the complete story because in the cell line that actually showed the best protective effect, we didn't see this upregulation in the MCL1, but this may be one mechanism. And then when we combine now MCL1 inhibition with this BCL2 inhibition, you can see that we can overcome this resistance that was induced. So in summary to this first part of the talk, so the adipocytes or co-culture with adipocytes reduces the maximum proliferation of AML cells and their viability, yet it enhances their maximum respiration. And this then protects AML cells from the effects of Vidasa and venetoclax. So these adipocytes have a bit of a dual role there. Now, when going back to the AML and its genetic pattern, you can see that in the AML cells, a lot of the mutations that lead to the leukemogenesis are actually mutations in epigenetic regulators. So many epigenetic regulators are affected there. One of the most famous one here, for example, is DNMT3A. But also the MLL genes and also many even of the other mutations involved here affect epigenetic processes directly. So my group has always been interested in epigenetic regulators in the context of AML. One epigenetic regulator that's not mutated, but that is found overexpressed in AML cells is called LSD1 or lysine-specific demethylase 1. LSD1 catalyzes the demethylation of H3K4 and the demethylation of H3K9, which is a bit of different factors because one of them is really assigned for heterochromatin. One of them is involved in the downregulation of gene expression, whereas the other one is involved in or marks active enhancers. So it's actually involved in getting genes expressed. It's an FAD-dependent monoamino oxidase, so it can be targeted by inhibitors, and it's been previously shown to be important in certain subgroups of AML. We therefore already during my time in Frankfurt got interested in the role of LSD1 and generated murine model systems to model this. The way we did this was by taking bone marrow cells from mice and then inducing leukemia in these bone marrow cells by retroviral transduction. And the two retroviruses that we use, or two combinations that we used, was one combination of FOXA9 and MEIS1, which are two oncogenes that induce then leukemic transformation. The other one is MN1. And when you put these retroviruses into these bone marrow stem cells, these turn into leukemic cells that you can grow in vitro. And when you inject them into mice, it gives rise to leukemia in the mice. So we could do this with a conditional knockout model and thereby had a model where we could very specifically switch off the LSD1. And when you do this, then what you can see is that proliferation is reduced. And the other effect that you're seeing is that the cells show differentiation, and in particular in this HOXA9-1 model here. You're seeing granulocytic differentiation. Granulocytic differentiation in the mouse model is always characterized by these donut-shaped nuclei here that you can see here. But when we look at those cells by flow cytometry, though, we see that this is not normal differentiation. This differentiation is characterized by both features of monocytes, but also by features of granulocytes. So a bit of a mixed type of differentiation. And so we wanted to understand what the mechanism was, and we did gene expression studies. And these gene expression studies then showed a lot more upregulated genes than downregulated genes, pointing towards a bigger role of this enhancer mechanism, really. And then the other thing was that when we then went into these expression profiles and used in silico prediction to predict which transcription factors contribute to these expression changes, we found that the biggest factors that contribute to these expression changes are two transcription factors, one of which is called GFI1, the other one is called PU1 or SPY1. And these we both found in the MN1, but also in the Hox-Mies model. And but there were also differences. And here, one of the differences, for example, is IRF8. We then did IP experiments showing that there was this or confirming essentially that there is this interaction of LSD1 and GFI1. And then looked in published data sets that where these GFI1 peaks are located. And actually many GFI1 peaks are actually co-located with these PU1 peaks. So what we think is that these PU1 target genes are regulated actually indirectly by the GFI1 or by co-recruitment. And two of these target genes I'm showing here where the peaks then are. You can see that the peaks are co-localized. And so the mechanism here is that LSD1 inhibition essentially takes out this downregulation of the PU1 target genes that is mediated by GFI1 and then leads to the upregulation of transcription factors contributing to differentiation. LSD1 can be targeted pharmacologically, and there are different types of inhibitors. Many of them actually target the coenzyme, which is the FAD, so they make a covalent binding to this coenzyme. And the other one is, or other ones are non-covalent inhibitors. So we were very interested at this point to study these non-covalent inhibitors because some of those were synthesized in a collaborating lab at that time. But unfortunately, these non-covalent inhibitors didn't exhibit the same effect. So whereas we saw differentiation with the covalent inhibitors, we didn't see it with the non-covalent inhibitors. So we asked the question if eliminating the LSD1 enzymatic activity is sufficient to induce the effect. And so we used an LSD1 conditional knock-in of an enzyme dead mutant to ask this question. And we could see that here, when you use the conditional knock-in, you're not seeing a survival advantage in the mice, whereas with the conditional knockout, you see it. So it's probably additional factors in addition to the enzymatic activity that plays a role here. LSD1 inhibition, when we look at that with one of those inhibitors, targets leukemic stem cells. So we then went back into the data and we got very interested in understanding how it targets leukemic stem cells. And at the same time, there were all of these publications coming out about the specific metabolic requirements of the leukemic stem cells. So we got interested in how LSD1 inhibition, or in which pathways LSD1 inhibition interferes with. And interestingly, we find a huge cluster of pathways that is upregulated here with LSD1, or changed here or altered here with LSD1 inhibition that's actually metabolic pathways. So when Kanwal joined the group about 2.5 years ago now, he got very interested in if we can target LSD metabolism by LSD, or if LSD1 inhibition targets LSD metabolism, and specifically then in the question, is a combination of venetoclax with LSD1 inhibition beneficial in that context? So again, going back to that seahorse machine that I've talked about before, when you study how the leukemic cells are targeted by the venetoclax, you can see that the oxygen consumption rate in these leukemic cells goes down when they are treated with venetoclax. So really these leukemic cells change their oxygen consumption rate. When you just block oxidative phosphorylation, when you just block mitochondrial oxidative phosphorylation, what you would usually see is a response of the cell that they increase their glycolysis, so that they increase their ECAR. The leukemic cells don't do this when you treat them with the venetoclax, but they still get more dependent on glycolysis, because when you make a ratio of oxidative phosphorylation, oxygen consumption rate, and ECAR, that ratio goes down. So the cells become more dependent on ECAR. Kanwar then studied with the same instrument, essentially, what happens when you do LSD1 inhibition. And using different inhibitors, he could show that actually the oxygen consumption rate, or this OCAR to ECAR ratio, actually goes up when you combine when you treat with the LSD1 inhibitor. So essentially, thereby showing that this indirectly targets glycolysis. So when we now look at this, that LSD1 inhibition targets glycolysis and venetoclax targets oxidative phosphorylation, this could be potentially synergistic. So we studied that in the leukemic cells, and really when you put together the combination, OCAR and ECAR both go down synergistically. And we then further wanted to understand what was the impact of that on the other metabolic processes in the cell. And we worked together with collaborators in Frankfurt doing MR spectroscopy of metabolites and sent them cells treated with this combination. And what you can see then is that there is in many of these metabolites that are important metabolites in particular in energy metabolism, that there is a synergistic reduction when you co-treat with with the LSD1 inhibitor, in this case, bomedemstat and venetoclax. So when you map this then on the metabolic profile of the cells, you can see that this is actually many different metabolic pathways, in particular in the TCA cycle, but also glycolysis in some of the side pathways here. So we then obviously were interested in the question if this is synergistic, and it is synergistic on the viability of the cells. This is also true in primary cells, and we're now studying these phenomena in vivo, and these experiments are currently ongoing. So hopefully soon we'll be able to report about some of those. Before getting to the end of the talk, I would now like to just briefly highlight another project that's going on in the lab. And this is really a project targeting immune escape in leukemic cells. So really going back to that question, how AML cells actually relapse if in the setting of allogeneic stem cell transplant. And the way this works is that leukemic cells are usually eliminated by an effect that we call graft versus leukemia effect. So the donor T cells recognize the leukemic cells through the HLA molecule and minor histocompatibility antigens, and then eliminate those. There is, on the other hand, the risk that these cells could recognize anything in the body and then cause a condition that we call graft versus host disease. So immunomodulatory strategies that could then shift these effects, so go away from graft versus host disease and enhance at the same time the recognition of the AML cells, could therefore be very beneficial. So Yu Yan, who is working in the lab on this project as a master's student, did a very nice review about this, where he then looked at different immune escape mechanisms, and many of them, when looking at them in detail, are actually involving epigenetic mechanisms, downregulating, for example, MHC molecules, but also downregulating for example, NK cell activating receptors, so that these then get less, or that the leukemic cells can be less recognized by the immune cells. So the question then was, can we enhance the recognition by LSD1 inhibition? And when we were looking at this, we wanted to see if we can find indications that they would enhance, that treatment with the LSD1 inhibitor enhances the immune recognition. One initial hint that we had that this could be the case is that many of these AML cells, when we treat them, upregulates actually one of the most important co-stimulatory molecules, CD86, which is a very important molecule in T cell activation. The other thing that we were looking at then was C2TA, which is the class 2 transactivator, which is the major regulator of the MHC class 2 complex. So when treating with the LSD1 inhibitor, a bunch of different AML cell lines, so actually most of those analyzed upregulated C2TA. And we then asked the question, does that also lead to an upregulation of HLA-DR, which is the most important, or which is one of the class 2 molecules in humans? So HLA-DR we can look at by flow cytometry. So when this is now here shown on the x-axis, on the y-axis, it's the CD86. So when we treat these cells with an LSD1 inhibitor, the cells upregulate CD86. When we then, here CD86, when we treat with the combination of interferon, which then further enhances MHC expression, we can see that in the presence of LSD1 inhibition, these cells then, more of these cells express the class 2 molecules, thereby essentially turning them into antigen-presenting cells. At the same time, it's obviously very important what cytokine milieu is close to these antigen-presenting cells. And one of the important cytokines here to enhance the immune response is IL-12, and one or quite a few, or 2 of the 4 cell lines studied here showed an upregulation of IL-12. We are therefore having ongoing collaborations with a group of also Dr. Bramson, and Hugo is also now in his master project getting more into treating or into investigating how the effect on the T cells, or how the effect of these cells is on T cells. And so what the experiments that he's currently doing is using a model system. This model system is a system that uses ovalbumin-specific T cells. This is the OT2 mouse strain. So it's a transgenic mouse strain for an ovalbumin-specific T cell receptor. And he's then incubating these T cells with the leukemic cells that have been pre-treated with the LSD1 inhibitor. So that would then show these effects that we've previously discussed. So, and preliminary experiments that he has done show that actually he sees quite a bit of an increase in cell death or a reduction in the leukemic cells after in the co-treated cells. So this is here in particular in the Hox-Mies model. The MN1 model doesn't seem to be as sensitive to that, but the Hox-Mies model is. And so what Hugo is currently mostly doing, or what Kurt is mostly interested in, is to look at what is the effect really on the T cell activation there. So do we see T cell activation in this model? So summarizing this third part of my presentation is that AML cells treated with LSD1 inhibitors exhibit signatures of activated professional antigen presenting cells. So they show an increased antigen presentation, increased co-stimulation, and lead to the production of, which then they also produce in pro-inflammatory cytokines, which then increases the responsiveness of AML cells to T cells. So the question or the long-term vision behind this project really is to use this in patients after allogeneic stem cell transplant. And one of the ideas obviously is to use this in patients who are relapsed. This could be done in the combination with the LSD1 inhibitor as well as venetoclax, and this clinical trial is actually is being brought on the way currently by collaborators in Miami. But it could also be a very interesting strategy to fight minimal residual disease cells after allogeneic stem cell transplant, because we know that when there's minimal residual disease after the allogeneic stem cell transplant, there is a very high risk for the patient to relapse. So fighting these or getting the immune cells, the donor immune cells activated against these minimal residual disease cells would be a very, or potentially very beneficial effect of this treatment. So this was also interesting for the OSCR. So they're now funding a project investigating this question, potentially leading up to a clinical trial where we have teamed up with colleagues in Ottawa, but also in Toronto, and hopefully we'll be very soon pursuing this also on the clinical side. But up to then, we still have a lot of work to do in the lab, and none of this work can happen without the team in the lab. So thank you very much to the team in the lab. We're all sitting over there. Thank you very much to all of you, and also to all of the collaborators and funding bodies. And with this, I would like to end my talk. And again, thanks for the opportunity to present here.
Thank you very much for a great talk. Do we have any questions for Tobias?
Okay. Jonathan?
What kind of data do you think you need to justify a clinical trial with LSD1 on top of venetoclax and I can't recall the drug was cytarabine or whatever it was?
Oh, okay. So for a clinical trial that would be trial in relapse patients, I think the question that I got a couple of times, why would you combine those two? Because those mechanisms have nothing to do with each other. So we have some data now that are very interesting in that context, in particular the data from Canvas that show that this is actually targeting a metabolic mechanism. So there is a mechanistic thing behind that. So this is kind of nice. We obviously want in vivo data, which we are currently trying to generate, or which we are currently working on with primary human cells. So we have this AML that I showed the data from, that is an AML sample that is engrafting in immunodeficient mice. So we will now do this co-treatment in this mouse model. And I think that would be a very important thing, because obviously when you're targeting both of these pathways, when you're targeting glycolysis and when you're targeting at the same time oxidative phosphorylation, one might ask the question, is this very toxic for normal cells? That may be some additional data that we need to generate as well. But the best way of doing that is obviously to do that in a living animal and still show that it lives after that.
When you implant your mouse lines, when you're showing that dose responsiveness to the relapse, could you use a murine syngeneic system to ask some of these questions if you think there's an immune component?
Yeah, we can. We are working on that as well. That's essentially the project that I want to pursue in the long run, and that Hugo is currently working on. So we are using this syngeneic model, the Hoxmies and the MN1 cells, which we'll put into the mice, and then we are currently trying to adapt this model so that we can use it together with the ovalbumin, or with these ovalbumin specific T cells. But obviously that could also be done in the model on its own, because maybe there may be some recognition in the absence of, but we irradiate these mice and they then get reconstituted with additional bone marrow cells so that they survive. So it's a bit of a complicated model to observe immune effects if we don't put back defined numbers of T cells. Potentially one of the other possibilities would be to use actually MHC deficient, or no, not MHC deficient, but MHC discrepant immune cells, or co-transplant MHC discrepant T cells, and see if their effect could be enhanced. That would also then answer the question if that enhances GVHD, because that's obviously something that we don't want to do. Right. In the long run, it may be the best way of looking at that question would obviously be a humanized mouse model.
You have a question in the chat here.
Okay.
Give me live audience some more time to ponder. Oh,
Yes.
who can see your message? Oh, nothing yet. Yes, Professor Ashcraft.
As we age, or both human and mice, well, the bone marrow is not replaced, but get more and more adipocyte in the bone marrow. So is that a correlation between like obesity or adiposity in the bone marrow and the frequency of AML or not?
It's not necessarily a function of obesity. So even a very fit, like not very obese person would have a high percentage of adipocytes in the bone marrow at when we age. Essentially, the calculation that the pathologist always says there is like about the percentage of adipocytes in the bone marrow is about your age, your numeric age. So at the age of 60, you probably have 60% adipocytes in your bone marrow. At the age of 80, you even have 80%. It's a question then in the context of these AML cells, because when the AML usually manifests, many of those adipocytes are actually pushed back or because of the growth of the AML cells. So in that setting of a primary manifestation of the disease, those adipocytes, it's a bit unclear what their role in that setting is, but when you then treat the patient and the leukemia cells are reduced or there's cytoreduction, then these adipocytes also would come back. And then when you're looking at those bone marrows that are after induction chemotherapy, there's actually a very high percentage of adipocytes. So in particular in that setting, there could be a big contribution to the resistance. But if that's also a contribution then to leukemogenesis, that's one of the reasons why these leukemias are more common with age. I'm not 100% sure. There are probably other factors that contribute more to that. Things like essentially genetic changes that are accumulated with age. And these things. Also, maybe some of the stem cell changes, because there's some data, in particular from the group of Connie Eves, show that our hematopoietic stem cells also change with age. So they get a more myeloid predominance, and they show some changes that could then, when you push them a little further with 1 or 2 mutations, lead to diseases that are maybe MDS, maybe even an acute leukemia.
Any further questions? On that last slide, you had some compounds up. Go to that last slide.
Which one?
I'm curious about something, because nobody else knows. One before this. Yeah. This, what are these here? That doesn't look like, so beside that, yeah, on the right side. Is this a separate?
So when you look at this thing,
Yeah.
the LSD1 inhibitors usually are always characterized by this cyclopropane ring here.
Yeah.
So that's the group that attacks the FAD.
Okay.
And then what they attach to the phenylcypromine, so the one that I have here, I can go back to that slide. I unfortunately don't have the structures of the other ones there here. So the phenylcypromine, which is the simplest one, which is originally been designed as an antidepressant and mostly targets monoamino oxidase, so the one that's needed for dopamine metabolism. So it's used as an antidepressant. And that is very simple, has these groups like, I would need to, I don't remember which other groups are there. You probably know that, I don't. But these groups have then been replaced by other groups here to make it more specific for the LSD1. And then what these other things are, why these
Yes. So
containing
are those groups
ones
that
are what their role is, if that's just a solubility thing or if that's
substitute out? Those are modifications of that core. Those get added to that main drug.
no, it's a separate. I think like that's the molecule,
Yeah.
and these are
That's what I'm wondering about. Yeah,
added in. I think
they're added in.
yeah, I think they are separate. I'll have to look again
Interesting, yeah.
what exactly that is. I think it's complex in some
I
way.
have no problem dominating this for a personal conversation between two of us. Anybody has any other questions?
Yeah, I think a lot that went into the design of this was to prevent actually penetration into the CNS, to be very specific for LSD1, but to prevent penetration into the CNS, because some of the earlier LSD1 inhibitor trials failed because of CNS toxicity. In particular, with one of the most potent ones that I have on the GSK LSD1, and GSK unfortunately had to cancel their LSD1 inhibitor program.
Yeah. So is there any like AML specific human antigen that can be lineage specific to be targeted for CAR cell therapy or something like that?
It's a difficult question. There are some. How specific they are, it's not clear. So what people have designed or used as targets, something like a CD123 or so, which is an IL-3 receptor, or CD33, which has been used as antibody target. They are not very specific for AML. They are on all myeloid cells. So using those for CAR T is probably not a good idea. There have been publications where they use some more obscure antigens, but how specific they are, then it's a little bit of a different question. One phenomenon that we have in AML cells sometimes is that we have co-occurrence of multiple markers, which are AML specific. So, for example, they sometimes have a co-expression of CD7, which is usually a T-cell marker on their surface. And then having a CD33 and a CD7 on the same cell, that could be something AML specific, but that's something that's a bit complicated to target maybe.
I don't think there's any further questions, so join me in thanking Dr. Tobias Berg